right. Great. Let's see, we have everyone and is coming. Great. All right. So without further ado, I think we can start this exciting webinar. On behalf of the ILA Young Epilepsy section, I would like to welcome you all to the webinar series. This is the second webinar that the ILA YES team is organizing. And I'm gonna give you a brief background about what is YES. YES is a wonderful organization that is fostered by the International League Against Epilepsy. It provides many opportunities for young investigators about networking, peer support, education, leadership, and many other support that you need. And in this context, the YES team has prepared a series of webinars that you will see for a couple of weeks now. Also, the YES team has a call for applications to lead a task force for advocacy, research, communications, and interactive media, conferences, education and publications, as well as membership. So make sure you go online and check all the opportunities that are available to you. So today it's my great pleasure to host an exciting webinar about high-frequency oscillations in epilepsy. We're going to be talking about their specificity to epilepsy, whether it's an accurate biomarker, and we're going to give the two sides of the coin. You're going to hear about basic science and clinical research. And also make sure that you follow the program that the YES team has prepared. There are many webinars. The next one is going to be about interictal activity and how it transitions to ictal activity. And with that, it's my great pleasure to introduce you to our speakers. Our first speaker is Dr. Laurent Seybani. He's a neurologist specialized in epileptology and sleep medicine. He's a postdoctoral fellow at UCL, where he's using intracranial recordings in people with epilepsy, and he's looking at the nice interplay between epilepsy and sleep entities and how this can modulate the risk of epileptic activities. Laurent, he's also involved in ILA, specifically the podcast, and he's also an associate editor for Brain Communications. Our next speaker is Dr. Julia Jacobs. She's the director of Alberta Children's Hospital, pediatric epilepsy program. She's a physician scientist working in epilepsy for more than 15 years. With many contributions in the role of high-frequency oscillations. In Calgary, she's also associate professor there and an adjunct professor at the University of Freiburg. And today she's going to be giving us the perspective from the clinical side. So with that, I'm gonna stop share my screen. And before I do that, I want to ask everyone to post their questions under the Q&A button, and we are gonna have a discussion at the end of the talks. So with that, stop share my screen. And Laurent, it's up to you now.
Okay, do you hear me well?
Yes.
Yes.
Yep.
Great. Well, thank you very much, Christos, for inviting me to this webinar. I'm very excited to have a very large audience also. So today I will discuss the mechanisms of high-frequency oscillation generation. And the idea that I would like to convey is how can we use our understanding of the mechanism of HFO generation to better understand their role as presumable epileptic biomarkers. I will go through a brief introduction of biomarkers in pre-surgical evaluation of epilepsy. I will then discuss the mechanisms of generation of HFOs. I will then briefly discuss the coexistence of pathological and physiological HFOs, And at the end, I will also discuss the mechanisms of seizure generation, and you will quickly understand why it is important to compare the mechanism of generation of HFOs with that of seizure. So epilepsy is a very frequent neurological disorder. It affects up to 1% of the population. And among these people, up to 30% will present a drug-resistant epilepsy. That is, despite the use of at least 2 anti-seizure medication, these people continue to suffer from epileptic seizures. And for these people, we can propose surgery. That is, we identify the brain region that generates seizures and we remove it surgically. But despite surgery, 5 years after the removal of the seizure onset zone or the epileptogenic zone, 50% of patients continue to suffer from epileptic seizures. And 10 years afterwards, it's even worse. Up to 60% of them continue to suffer from epileptic seizure. So there is a gap in clinical knowledge here. We need to have better biomarker to precisely identify the region of the brain that should be removed to optimize the prognosis after surgery. And HFO could be one of these biomarkers. So pre-surgical evaluation of epilepsy basically means surgical planning. Which part of the brain should be removed to optimize seizure freedom? There are several brain areas in this topic. We have the structural lesion, that is the region of the brain that appear with a lesion on the MRI. There is the irritative zone, which is the region that generates intraictal epileptiform discharges. We have the ictal network that encompasses the regions recruited or that maintain seizure activity. And we have the seizure onset zone, which is the region of the brain that we think generates seizures based on the EEG, based on other evidence from clinical evaluation. And there is an overlap between these two, between all these different regions, but the overlap is not 100%. So now HFOs could be an epiphenomenon of epileptic activities. And in that case, I would say they are an okay biomarker, a useful biomarker, but not the perfect biomarker. Or they could share generative mechanism with seizures. And in that case, they become a great biomarker, right? If they share the same mechanism of generation, then we can expect that they will be very specific to the region of the brain where seizure starts, and they become great biomarker. So what are they? Here is an example of an HFO recorded in a mouse model of temporal lobe epilepsy. In black, you have the raw data. In gray, you have the filter data. You see very well the oscillation in a spindle envelope, if you want. And high-frequency oscillation, HFOs, can be divided into ripples between 80 and 200 hertz, fast ripple between 200 and 5 to 600 hertz, and very high-frequency oscillations. I won't have time to discuss this entity that was only recently described. So what are the mechanisms of generation of HFOs? At least what do we know so far? A few historical dates that I think is important to know. In 1992, the first identification of ripple by Buzaki and colleagues, they actually do not use the word ripple in their paper. I recommend you to read this paper. It's very exciting to read the very first paper where they see a ripple in the hippocampus of an animal. A few years later, they use this time the word ripple, and they predict the role of this ripple for memory formation. And in 1999, ripples are identified in humans and fast ripple in humans with epilepsy. Now, what's interesting with ripple is that if you compare the activity of excitatory cells, pyramidal cells and interneuron, it's very much different during ripple. Pyramidal cells tend to fire just one or a couple of times during the ripple. For example, this one fired at the end of the ripple, while interneurons fire more systematically along the duration of the ripple. This is a very nice figure where you see that this interneuron fires at each trough of the oscillation. So they have a different firing pattern during the ripple. Interneurons have a central role in the generation and in the maintenance of the ripple. They fire, as I said, along the duration of the ripple. The current model is that interneurons, such as basket cells, for example, contact pyramidal cell on the soma, and they lead to perisomatic inhibition of these cells, and these inhibitory postsynaptic potential add up and make the fast ripple. So the ripple. So ripple are believed to be to reflect the summation of inhibitory postsynaptic potentials. Now, while interneurons are important to maintain ripple, it seems that pyramidal cells might be particularly relevant for ripple initiation. In this study, you can see in vitro a ripple, and inhibitory postsynaptic current and excitatory postsynaptic current. In here, you can see that the blue line, so the excitatory current in the pyramidal cell, increased before the red line, which is the inhibitory current. So excitation of pyramidal cell anticipate inhibition of pyramidal cell. So while interneuron might be important to maintain the ripple, actually activation of pyramidal cell precede the inhibition of these cells. But if you inject a GABAergic antagonist, such as gabazine, as it was done in this experiment, you see that the amplitude of the ripple is decreased. So interneurons are still very much important during the ripple, but the initiation might be dependent upon excitatory cells. I won't go further on the mechanism of ripple formation. I'd like to spend more time on fast ripple now. So fast ripple are oscillation between 200 and 600 hertz. There are some neurons that can fire up to 800 hertz, but in general, neurons fire at much lower frequency. So how can we observe oscillations at such high frequency? Well, there are two possible mechanisms. One hypothesis is the hypersynchronization hypothesis, and the other one is the emergent oscillation. In the hypersynchronization hypothesis, the idea is that all neurons fire at each peak or each trough of the fast ripple. If you put a line on each action potential, you see that it aligns with the trough of each trough of the fast ripple. And in contrast, for the emergent oscillation, the hypothesis is that each neuron do not fire at every trough of the oscillation, but altogether, if you look at the emergent oscillation, you have basically the same pattern, because although each neuron do not fire at each trough, there is a neuron that fire at each trough. So in contrast to ripple formation that depends upon interneuron perisomatic inhibition, fast ripple seems to be much more dependent upon presynaptic activity, so axonal activity. It's a summation of action potential. Now the question is, in the epileptic brain, how this fast ripple can be generated. So if you take a normal brain, this is in vitro recording in non-epileptic animal, and here in epileptic animal. If you activate a cell, you will see that there is a second spike that occurs at a reliable timing since the first trigger. In epileptic animal, there is a lack of reliability in the delay between the first and the second spike. And this could be consecutive to structural damage in the epileptic hippocampus. One possibility for the emergence of fast ripple is this lack of reliability. Think of the fast ripple as a sort of pathological version of ripple. If you have a ripple here, you have the observed oscillation is made of the firing of different units at different time points, and eventually you see an oscillation within this frequency band. But if you have a lack of reliability, the second spike, for example, will fire at a closer delay since the first spike, the third as well, this one as well, but then you have an increased delay here and an increased delay here, and eventually the frequency that you will observe is about 2 times faster than the initial frequency. So that could explain why in control animal we have one single peak of activity when we look at the time frequency of ripple, while in epileptic mice, we have a sort of harmonic peak 2 times faster than the original oscillation. And that's explained by the fact that you have an out of phase, a lack of reliable delay between action potential of successive neuron. So which one is occurring in the epileptic brain? Well, it's difficult to assess, but this study tried to answer that question. What they did was to plot the contribution of high frequency component in the spectrum. That is whether the fast ripple is mostly made of 400 hertz or 600 hertz, and they correlate that with how dispersed is the frequency spectrum as measured by the entropy. So if you plot the entropy of the fast ripple and the fast ripple index, which is the contribution of fast activity in the fast ripple, you have basically two distributions, and each of them reflect two different kinds of fast ripple. The negative correlation reflect pure oscillation, so the hypersynchronization mechanism, and the positive correlation reflect more the emergent oscillation. I won't have time to go into the details of this, but we can discuss that if you have questions. But basically what's interesting is that depending on the region where they recorded, they found that the mechanism could be different. In the dentate gyrus, the analysis suggested that the hypersynchronization mechanism might be more relevant, while in CA1, the emergent oscillation hypothesis might be more relevant. So depending on the brain region where you record the fast ripple, the mechanism of generation could be different. Fast ripple have also an important impact on the epileptic network. I wanted to discuss two studies here. In this study, we recorded awake mice with epilepsy using scalp electrodes. So we had 31 electrodes distributed equally over both hemispheres of the epileptic mice. We injected kainate in the left hippocampus, located more or less here, and we recorded fast ripple across different time points after kainate injection. So before kainate injection, we have a lot of fast ripple recorded over the somatosensory cortices, and this is actually also known in humans. But after kainate injection, you see a massive increase of fast ripple in the region of the epileptic focus, that is in the left hippocampus. Interestingly, pathological fast ripple seem to have a slightly lower oscillatory frequency than physiological fast ripple. And the other important information is that, well, of course, it is true that the fast ripple increased massively in the left hippocampus, so in the epileptic hippocampus. You also have fast ripple that appear outside of the initial epileptic focus. Fast ripple are thus important for the epileptic network. Another important studies about this is this study in human, where they use microwires that are able to record single unit activity. And what they found is that locally fast ripple induce an increase in firing rate. The color scale is the firing rate. It's blue when it's decreased and it's yellow when it's increased. But globally, as you can see on the average, there is a decrease of firing rate. So locally, fast ripple leads to an increase in activity, but globally, outside of the region where fast ripple were generated, there is actually a decrease of activity. So fast ripple have large-scale impact on the brain. Just to summarize this first long part, physiological ripples are coordinated by interneurons. That's a core mechanism for their expression. Pathological fast ripple are more likely to be the product of summated action potentials. There are two mechanisms that can coexist for fast ripple generation: hypersynchronization or the out-of-phase activity. And fast ripple can emerge outside of the initial focus. I didn't talk about the impact of sleep or the sleep-wake cycle on fast ripple generation. We know that slow wave activity during sleep impact high gamma and neuronal firing. So it's very likely that the mechanism of generation of fast ripple could differ between sleep and wakefulness. You can have a look at this review where we discuss some of these mechanisms. So now, coexistence of pathological and physiological HFO. Initially, it was thought that the frequency, the oscillatory frequency, could differentiate pathological from physiological HFOs. And pathological HFOs were thought to be of higher oscillatory frequency. But it is now known that this boundary is not that clear. And so a question that I think is very important is whether within the same frequency band, for example, ripple between 80 and 200 Hertz, can we see ripple that are made of physiological mechanisms and ripples that are consecutive to pathological mechanism. And again, the answer is not solved in the literature, but I wanted to share with you this interesting paper that I tried to look at that. So here, what they used is human tissue with a pro-convulsant. It was an in vitro study, and they identified pre-ictal discharges and intra-ictal discharges associated with ripple. The difference between pre-ictal in red and intra-ictal in black was the amplitude of the intra-ictal discharges. And they looked at the ripple associated with these discharges. What they found is that the intracellular activity in pyramidal cell, the ring ripple associated with these discharges were different for intra-ictal and pre-ictal discharges. For pre-ictal, it was mostly a depolarization, as indicated by this positive change in regard to baseline, while for intra-ictal ripple, some of them were associated with a hyperpolarization, and others were associated with a depolarization. So this suggests that you can indeed have ripple generated by different mechanisms depending on where you sampled them in regard to seizure onset. Now, mechanism of seizure generation, I think, is an important topic because, as I said before, HFOs would be a particularly good biomarker if they share some mechanism of seizure generation. The problem is that the etiology of epilepsy is very variable. You can have structural, genetic, infectious, metabolic, immune, and unknown mechanism for seizure generation, which already suggests that HFO might probably not share all the possible mechanism of seizure generation. For example, here they recorded with microwires the onset of seizures in the human brain, and they showed that the activity of neurons is very much heterogeneous across different neurons. However, across different seizures, it seems that the same cell is reproducing the same pattern. So on the one side, the heterogeneity of activity seems not exactly in line with what we know about the mechanism of generation of HFOs, but the fact that the same cell do the same thing across seizure seems more likely to reflect the activity of cells during HFOs, where you have a sort of standardized pattern of activity during the oscillation. Another important point is that you have different types of seizure onset. You have seizure onset that are qualified as low voltage fast activity. So you see in these two electrodes, you have fast activity and low amplitude. This is another example where you have low amplitude fast activity. And the other seizure onset type is the hypersynchronous type. So you see you have a spike here and then another one here. So it's high amplitude and periodic discharges like this. So two different types of seizure onset. And what's interesting is that if you activate interneurons with optogenetic, you're more likely to elicit a low voltage fast onset, while if you activate excitatory cells with optogenetic, you're more likely to trigger these hypersynchronous onset of seizures. So this to show you that you have different type of seizure onset, and these different type of seizure onset reflect different recruitment of cells at the beginning of seizure, which again suggests that the mechanism of seizure generation is probably not always reflected in the generation of HFOs. So don't take me wrong, I'm not saying that HFOs are not correctly reflecting what's going on during the seizure. I'm saying that the mechanism of HFO cannot reproduce entirely the very complex and various mechanism of generation of seizures, which could explain why they are not the perfect biomarker for pre-surgical evaluation of epilepsy. So the take-home messages would be that you have different mechanisms that lead to ripple formation. For ripple, the perisomatic inhibition of interneuron is very important. For fast ripple, the presynaptic activity seems to be more important. Fast ripple can emerge outside of the serum onset zone along epileptogenesis, at least in a mouse model of epilepsy. And very interestingly, for the same entity, that is for ripple, within the same frequency band, you can have different mechanism leading to that generation. Some ripple will be associated with either a hyperpolarization, so inactivation or activation of pyramidal cells. Other kind of ripple will be associated with an activation of pyramidal cell. So it's a very heterogeneous activity at the onset of ripple. I'd like to thank all the collaborators on the different projects that I presented here, my fundings, and you for your attention. I'm looking forward for the talk of Julia Jacobs and for your questions afterwards. Thank you.
Great. Thank you so much. Thank you so much, Laurent, for the exciting talk. And now the floor goes to Julia Jacobs.
Julia, I think you need
Yeah,
right click on a slide.
I can't even find, give me a sec. I have to share
Sure.
again. So again, it has this problem we had earlier. I don't know why.
And yeah,
Like
full
this.
screen.
Yes, wonderful. Can you see me all? That's great.
Yeah.
Welcome, everyone. I'm happy to talk about high-frequency oscillations from the perspective of the clinical application. You all learned a lot already about the basic science between high-frequency oscillations. And I think I want to first thank the Yes section that they invited us to give a talk on a very important day, because as many of you might be aware, today is actually Purple Day for epilepsy awareness. So I wanted to start and dedicate this talk to all of the many people who are celebrating and acknowledging Purple Day today, including Megan Cassidy, who is the Canadian founder of Purple Day, and just say, like, all of the work that we are doing is really, we are doing it for our patients and in hope that the world understands epilepsy better at some point. And without further ado, I'm going to go ahead and talk about high frequency oscillations. Basically, the learning objectives and aims for today are, I'm going to quickly talk about in a clinical setting, what are HFOs and how do we identify them, then clarify what value they actually have to identify epileptic tissue and guide surgical decisions, and then summarize what current challenges we are actually facing when we want to use them in a clinical content. You've heard all this already from my previous speakers, so I'm going to be very quick, but basically we have the different HFOs, and I think one of the important concepts in clinical application of HFOs is that we actually now know that it might be less relevant to always separate ripples, fast ripples, or even very high frequency oscillations, because there's a significant frequency overlap when we look at intracranial or scalp clinical recordings. So for the rest of my talk, I'm going to mostly talk about high frequency oscillations in general. And I think the other really important thing about clinical use of HFOs is that from the time when I worked more than 15 years ago, the first time on high frequency oscillations to now, we know that they are not limited to the single cell recording microelectrode, but they can actually be seen all the way to scalp EEG, and that of course has a very important clinical implications. Christos, who's our host today, organized this wonderful workshop on high frequency oscillations in New York last year. And I think one of the things that we learned from that is that even if you put 100 experts in a room, we don't always agree on how exactly an HFO looks. And HFO identification is still a big challenge when we talk about clinical use. But what we can also say is that most EEG amplifiers that you use in a daily clinical setting can record now with sampling rates about over 100 hertz. So you are able to actually record HFOs. We have to choose the right filters. And in general, the rule is use an FIR over an IIR filter because FIR filters have less ringing with them. And then, of course, most importantly, you have to choose and understand the identification methods that you want to use. And this is where we come into talking about detectors. If you want to use HFOs in a clinical setting, you cannot do what I did as a postdoc and spend hours and hours on visually seeing them. So we have to agree on what detectors are reliable. This is from a summary paper that we have prepared where we contrasted most of the existing detectors with each other. And the summary was basically that there's a large number of established HFO detectors. Most of them work best in the data they are trained on. Some of them have specific features. So, for example, you can contrast this baseline activity with spike or get a spectral analysis. And you should be aware what they're designed for and use them for what they're designed for. So, if you want to look at scalp EEG, you might have very different decisions on automatic detectors than if you look at intracranial EEG. Now, moving on to the clinical relevance of high frequency oscillations. So what we are going to talk exclusively again today about is really the localization value. So does it find epileptic tissue? What is the overlap between the epileptogenic zone and the HFO generating zone? But there is a whole other thing which I think HFOs can be interesting for, and I just wanted to mention here, and that's can it measure disease severity? So what's the risk of recurrent seizures? Do I treat successfully? Is that a patient with a poor or good prognosis? All of these things are what we would call seizure propensity or severe disease severity. I'm not going to talk about it today, but there's a lot of papers on this as well. Moving into the first big question or aim for learning for today. And this is, can we use HFOs for pre-surgical workups? And when we talk about this, what you can see here is basically the different levels of evidence. We first wanted to see, do HFOs identify the seizure onset zone? Then we moved on, do they identify whether someone is going to be seizure-free? And we did this in retrospective data, prospective data, and then individual patients. And then, of course, the highest level of evidence we can ever have in medicine are randomized controlled trials. And as always in medicine, we have a lot of data on the weak evidence and much less data on the higher evidence. And all of this is following a very simple hypothesis. If we want to say that HFOs are good biomarkers, we have to say when we remove them all, the patient is going to be seizure free. If we don't remove them all, the patient is not going to be seizure free. And this is old data, but I just wanted to show you, we did a meta-analysis on all the existing studies. And as you can see here, both for ripples as well as fast ripples, the evidence beyond zero is there, but it's actually not super strong in the retrospective data. And I think prospective trials are, of course, always higher level. And this is the first multi-center study which we did, where we basically, at the time of the implantation, used the HFOs to predict what will be post-surgical outcome 1 year later. And we used data from Freiburg, Montreal, and Los Angeles. And on the group level, so this is this level here, prospective group level, you can see that we had significant differences between the patients who did poorly and the patients who did well. Unfortunately, this didn't work out for the individual patients. So we had patients like this one who had an angle outcome 1 and were seizure free. But as you can clearly see by all these red dots, the majority of the HFOs in these patients were not removed. And there was another study from Switzerland who did something very similar at the same time. And this is the pooled data of both of these studies. And all the numbers in green are those where we predicted correctly. But you can also see that in 27% we had a wrong prediction. And in 14 patients, that's the one down here, we would have actually made a bigger resection than necessary to achieve seizure freedom, because they achieved seizure freedom despite not having all their HFOs removed. Of course, you can say now, okay, HFOs don't work, but we didn't want to do that. So we moved on into larger multi-center prospective trials. And this is now a collaboration between many different epilepsy centers. And as you can see, we now have 146 data sets that were used. But again, what you can see here is that there's green and red areas, and there is a substantial number again of patients in which the prediction did not work. And again, you can see that the odds ratio is actually not so great, especially for some centers in regard to predicting the outcome correctly, depending on the resection size. We can also always ask, do we look at the right biomarker? What you've seen in the past, we're always looking at ripples and fast ripples alone. This is another larger multi-center trial in which we were involved. And they actually showed that, again, you see there is not a perfect distribution, but for most of the sites, looking at spike ripples was actually more successful in localizing the epileptic zone than looking, for example, at the ripples or the fast ripples alone. Despite this, I would say, a bit discouraging results, the group from Utrecht has been super courageous in my perspective, because they said, and they set out and went for a randomized control trial. That concretely means that where HFOs are located will determine where the resection zone will be in this patient. And they used intraoperative recordings. And the interesting part of this is that they set it out to be a non-inferiority trial. What this means very concretely is that HFOs did not have to perform better than the gold standard, and in this regard, the gold standard was spikes, but they just had to be not worse than spikes. And still, unfortunately, this trial failed to prove that, because as you can see here, 67% of the patients in the HFO guided group and 90% of the patients in the spike guided group became seizure free. So why did this happen? And I think it's a great lesson on randomized controlled trials in epilepsy surgery. Because of the randomization, when they did post hoc analysis, what they could see is that accidentally the HFO group had much more patients with a poor prognosis pathology than the spike group. So it might not actually be related to the biomarker, but more by the random selection of the patients. And what they then did was they corrected for this, and they also corrected for temporal versus extratemporal lobe cases. And when they did that, they actually could show that HFOs are similarly good performing than spikes in the extratemporal lobe epilepsies. So I'm moving on to the last part of my talk, but not the least, because I think what we can say is that the trial of Maike Zielman shows that we somehow fail with our HFOs. So my last question for today will be, why do we fail? Or better to say, what should we do to better understand how to use HFOs as a biomarker? And I think I have given you 4 things here, which I'm going to give you a little bit detail on the 2 things. One of them is limiting spatial sampling in intracranial EEG. Do we actually look at the right biomarkers? So do we do ripples, spike ripples, fast ripples, and so on? Are HFOs actually very focal biomarkers, or are they, like spike and epilepsy, a network phenomenon? And last but not least, how do physiological HFOs influence our results? And I'm going to start with the spatial sampling because I think you all know if we look at intracranial EEG, we basically have a black box in the brain everywhere where we don't plant an electrode. And this means that we might have a patient where all the HFO seems to be removed and we are predicting a good outcome. But of course, who tells us that there's not HFO somewhere else in the brain? And this is where I think the news about scalp EEG is really important because, of course, if we could have a scalp EEG with the intracranial EEG, we might learn a bit about these black box areas. And the very early studies that I did on this were like looking at simultaneous recordings of intracranial and scalp EEG and proving that what we see intracranial as an HFO actually has a scalp correlate. Because many people were saying, well, all the fast activity you see on the scalp is actually artifact and HFOs cannot pass the scalp barrier. What we then could also show is that when we have widespread occurrence of HFOs on the scalp EEG, this is a negative predictor for outcomes. So if more than 4 channels had HFOs on the scalp, this was bad. And last but not least, we also could show that the classical EEG is actually undersampling again the scalp. So having high density EEG actually improved the HFO recording. So is that the solution to all? Maike Zielmans went ahead and looked at all these scalp HFO papers that are currently out there a year ago. Until now, there's probably much more. And basically, what she could show is that there is again a lot of studies which actually link scalp HFOs to epileptic areas, but there's also a huge inhomogeneity of how the methods were chosen for these studies. So the biggest take-home message at this point, I think, is that the methods that we are using in scalp EEG are very variable, and we don't have really good prospective data which tells us whether a scalp HFO can be a solution for this surgical setting. Now, talking about maybe we don't look at the right biomarker. There was this provocant paper by Rory et al. In Annals who actually said that spikes are better than HFOs. And especially spikes plus HFOs are better than HFOs alone. And the main reason for this is that fast ripples are very focal and linked to the seizure onset zone, but they lack sensitivity. And then on the other side, spikes plus HFOs were the best in all patients, but there were a couple of biomarkers, or like there were some patients in which certain biomarkers work better than others. And of course, that's always a problem if you work in a prospective way, because only retrospectively you will know which biomarker is best for an individual patient. I also wanted to highlight, and that's a bit like in consequence to the talk we've just seen about like what are HFOs and what's the basic mechanisms, that we don't still understand completely how spikes and HFOs are linked with each other, and that there might be a value of looking at the combination. This is the study that we did in Freiburg with simultaneous micro and macro electrodes looking at multi-unit activity. And what we basically could show is that if you have a spike that has an HFO associated, the firing pattern with a strong increase prior to the actual spike was very different from those spikes which did not have HFOs associated. So I think we are talking about two different entities, and not every spike is the same. And this, of course, comes very much into the center where I trained in at the Montreal Neurological Institute, because there was Theodore Rasmussen already many, many years ago, who had this idea that there might be red, more vicious, and green, less vicious spikes. And in my world, of course, I would like to call the red ones the one with HFOs and the green ones the ones without HFOs. And I think we can conclude by now that looking at a combination of biomarkers, spikes plus HFOs might be one way to find the most epileptic spikes, to exclude physiological HFOs, and to facilitate identification. But again, it's one of the questions and one of the possible explanations why we fail. The next topic is focal versus network activity. And I think here I just wanted to highlight what this means and my idea with the hypothesis I've guided you through. So we've always said, like, if I remove everything in red, the patient is supposed to be seizure free. If I don't remove everything, the patient remains to have seizures. But what if not all of this red are actually areas that generate HFOs, but some of it is propagation? And you know that really well from the spikes that we sometimes have widespread spike areas, which doesn't mean that the spike is generated over all of these. So this would really change our hypothesis because it basically means that I have to find the generator and separate it from the propagation areas to predict whether someone gets seizure free. Because in this scenario, I only have to remove the generator and not the yellow triangles to make someone seizure free on the long run. And there have been some efforts here from the group from William Stacy about trying to find out what are the network dynamics of high frequency oscillations. And what you can see here is actually very similar to what we see in spikes. You often have HFOs which are visible at the same time point over a wide area of channels. And what they did also additionally in their study is that they said we want to separate patients who have a high. So sometimes you can use the seizure onset zone to say, do we really remove the seizure onset zone? So does this patient ad hoc have a likelihood to be seizure free, or do we go for palliative resection? And they decided to only look at those patients who had at least 80% of their seizure onset zone resected to have a primary good prognosis in their surgery. And then they looked at these network changes. And what they could show is that when you look at not only the HFO rate, but actually the connectivity within the network, then you are more likely to actually predict properly. So this is 28 patients, 18 had a class outcome. They combined the network centrality, so where the HFO was generated, how central it was with this and the seizure onset zone in their predicted model. And then they had 13 correct predictions of these 18 and 4 where the prediction was not correct. Again, not ideal, but maybe already a move towards the right space. Last but not least, you have heard from the data that Buzaki has shown and that we've discussed in our first talk that HFOs are not all epileptic. We know for a long time that there are HFOs or ripples and fast ripples which are involved in physiological activity. And if we come back to the model again that I've shown you, this is our problem. We might have areas where there's only epileptic HFOs. There might have areas where there is mixed epileptic and physiological HFOs. And then we have these green areas which are mostly physiologic. And of course, you do not want to resect the areas over which there's a lot of function and not a lot of epilepsy. So how do we account for that? And unfortunately, HFOs look the same. It's a bit like salt and sugar. They're both white and grainy. Doesn't mean that they have the same taste, and same for the HFOs. So there's a couple of principles we can use. People looked at the characteristics of HFOs, so amplitude, frequency, length. They looked at background activity, like is it very oscillatory or very suppressed. They looked at different brain regions with the idea that maybe HFOs, physiological HFOs are predominant over certain reasons. And then they looked at co-occurrent with other events. The very early data suggested a simple change by characteristics, so saying all ripples are good and all fast ripples are bad. Unfortunately, and I think you've seen that in the data we've shown, this data, which was taken by the early group Staba and Bregan in microelectrodes did not prove to be right when we started working with macroelectrodes. So in our daily recurrence, not every ripple is physiological and not every fast ripple is pathological. So we have to get more sophisticated when we talk about HFO characteristics. And I think this is a very nice paper for one suggestion where they looked at how stereotyped high frequency oscillations were. So how consistent are they in their different features, amplitude, frequency, and so on. And they could show that when they are very stereotyped, so every HFO is very similar, they are less likely, they are more likely linked to the seizure onset zone and less likely physiological. We've also done efforts where we looked at electrical stimulation in intracranial EEG and said, like, do ripples look different when they are over a region where we have a functional response to stimulation compared to ripples that are in the seizure onset zone? And as you can see here, there are rate differences and there are also differences um, in regard to different features of the HFOs. But the problem is that I think we often have areas where both epileptic and physiological HFOs co-occur, and that makes it difficult to separate the two. But still, I think there is an importance of us working on more atlases who actually show the distribution of spontaneously occurring physiological HFOs, like this atlas from Birgit Frauscher, because it will allow us to know where physiological HFOs are very dominant and subtract those from the areas where we think the epilepsy occurs predominantly. And as you have learned in the introduction, I'm a pediatrician and quite passionate of making sure that we don't only look at this in adults, but also in kids. So one of the newest efforts of my group is actually to develop a physiological atlas of brain maturation for physiological HFOs. And this is what you can see here. We looked at 122 healthy children, and there is a substantial change over the time. You can see that HFOs increase in their rates until around 3 to 5 or 6 to 10 years, depending on the region, and then they actually come down again afterwards. And you can also see that there were significant changes in distribution. We saw them for the age groups, but also for the brain regions, and we hope that this can be used as a control if we want to identify clearly abnormal HFOs in the future. So to summarize for these physiological HFOs, I think and I've just shown you a few examples today, but there have been studies, a lot of studies, which try to find what are the most pathological ones. And I think we've spoken about spikes plus HFOs. I wouldn't know why a physiological HFO would choose to hitchhike on a spike. So I'm quite sure that these are abnormal. We have also stereotypic HFOs we spoke about, fast ripples we spoke about. And then on the other hand, we have some features which might identify physiological activity. So for example, HFOs at the peak of a slow wave in sleep, or HFOs plus sleep spindles, because why would an epileptic HFO run on a healthy sleep spindle? So I think we have some ways to separate these, but there we need more atlases, more combined detectors, and more functional testing to actually separate this clearly. On the last note, I want to highlight as well that we know that HFOs might not be completely epilepsy specific. And I think that is something we have to acknowledge, and we are just now learning more about. This first study here, for example, highlighted that neuroinflammation might have an influence on how often we see high frequency oscillations. And then, of course, we have this wonderful study here by Christos talking about high frequency oscillations in animal models of Alzheimer's, and we might have some human data as well in the future. And this is clearly not epilepsy linked, but you might also have HFOs even if you don't have seizures in Alzheimer's disease. To summarize, there is a strong link between epileptic areas and HFOs. The prediction of outcome in individual patients, however, is still unpredictable. It works in some, it doesn't work in all. A better understanding of HFO characteristics and networks interactions is needed. Physiological HFOs can impact data interpretation, but also provide new information. So we are just learning more and more about this. And HFOs might not be as epilepsy specific as we think, and that's also a future area we should work on. If you are interested in working on HFOs in a clinical spectrum, I think my most important take home message is there's not a one shop toolbox. You can't go into the supermarket and say, I want to analyze HFOs, give me a toolbox. So please, I think the most important way is understand what you're doing, understand the detectors you're choosing, understand how to identify an HFO, and then hopefully you can use them to provide additional information for your patients. And with this, I would like to thank my whole clinical and research teams, my funders, and of course you for your attention.
Great. Thank you so much, Julia, for the exciting talk. I think now it's time to move into the discussion. We have a couple of minutes. The first question is if you could give some tips about the technical aspects of picking up HFOs during epilepsy surgery, or a paper that people can look at. That's the first question.
Sorry, like what to pick what?
If you could suggest some technical aspects and filter settings to pick up the HFO during epilepsy surgery.
So is
And
that in intraoperative or like in chronic recordings prior? I can say a sentence to both. So I think if we are talking about the classical intracranial setting, then I think there's two tips. First of all, even if you have an older software, you often can be able to increase your sampling rate, and your software should have an ability to check this out. If you open your sampling rate, you might then start seeing noise that you're not aware of before. So, we have interference from cardiac monitoring, sometimes from televisions in the hospital rooms and stuff like this. So you should have a double check, how good is my data now? And then for the intraoperative room, I am citing Maike Zielmans now because I don't do this regularly, but I had a lot of discussions with her about it. So she suggested that you have to test every single amplifier you use in every single OR you're going into, because there's a high noise variability. And then there's two more important things. You have to make sure that your electrodes have really good contact to the recording surface in the intraoperative room. And if your patient gets a sedative which suppresses epileptic activity, you have to wean this so like most centers reduce, for example, the propofol during the HFO recordings. And the last and probably funniest comment I got from Michael was the most important is that you need a student who chats up the surgeon, because neurosurgeons are not very patient people. Sorry to all the neurosurgeons who are in this listening into this now, but you need a quiet recording environment. So you should make sure that you have a clear agreement on how much time you reserve for recording HFOs, and it should be at least 10 minutes in the operative recording so that you have reliable and stable data.
Great, thank you for this. So the next question is for Laurent. In the LFP, you saw that for the out-of-phase firing, out-of-phase hypothesis, why would it be that there is harmonic frequency? Wouldn't it be that there is a wide range of frequencies in the HFOs that are being produced under this mechanism?
Yeah, it's a good question. I would say so I use the word harmonic to say that there is a jump in the oscillatory frequency. If you look at the frequency of ripple and then fast ripple, there's a decrease in between these two peak of oscillatory frequency. Why it's not a continuous spectrum, I actually don't know, and it might be related to how the network is constructed, how the related to the refractory period of neurons and things like this, but exactly why, I don't know. However, if you look at the different papers, if you look at the papers by, I think I discussed it, Ibars from when he was at Menendez de la Prida lab, different model. I have some slide if you want, I can share. You always see this jump between the frequency band of ripple and fast ripple. So there must be some, I think, structural constraint that make this jump and not a continuous oscillatory frequency.
Great. So in the same, let's say, theme, it would be interesting to discuss whether there are ways that we can probably block HFOs. Are there any ways, like pharmacologically, that can target the mechanism? How good is the evidence that we can, target those oscillations?
Well, there is a very nice paper by Buzaki and with Girardo in first author where they detected ripples online, and then with don't remember if it was electrical stimulation or optogenetic, but they stopped the ripple while the ripple was ongoing, and they could show that this disrupt memory. For fast ripple, I didn't discuss that, but there are some evidence suggesting that gap junction are important for coordinating neurons during HFO. So gap junction are electrical synapses. They are physical communication between axons. And there's one study showing that if you put halothane, which is an anesthetic, that also is also an inhibitor of gap junction, you can decrease the rate of these fast ripple.
And is there any evidence from anti-epileptic drugs that could be more potent? Anything from?
I don't think we have. So I think the biggest challenge with anti-epileptic drugs is that we have some from chronic epilepsy models, but they wanted to more like look on the long run. It's not an immediate interjection, and I think there's data on levetiracetam, lacosamide, and some of the classical anti-seizure medications, also steroids, and they all show that if you give them the HFO get reduced, but it's not a mechanism specific action, right? And the same we have from looking at a lot of data by now for scalp HFOs. I think it's excited because what we see is that medication, that the HFOs actually are reduced when medication is successful. So we can see, if patients with infantile spasm syndrome, for example, successfully get steroids, their HFOs go down, while they don't go down if the patients don't respond to treatment. So that's a very clear clinical correlation. And I think what many people don't know is that this is actually not true for epileptic spikes. There's a lot of clinical data which shows that even if we treat successfully, spikes stay the same rate. So it's actually, this is like exciting about the biomarker HFOs, but unfortunately, I don't think it's any way mechanistic. It's really an observation of suppressing epilepsy activity rather than saying we are targeting HFOs, right? It's just a reflection.
Yeah. Thank you for this. And we have more questions. I think we're gonna do 2 more questions possibly. So I think that was also discussed, but I think if you could comment on whether what is the best way to differentiate pathological from physiological HFOs. I think that is probably, there's no consensus, but they suggest whether there is combination of duration, frequency, but amplitude, anything else that you would think maybe would be helpful.
So, I mean, the core problem of this question is that in the area where the epilepsy is generated, we might still have some physiological ones and we're not aware of it, right? But it might also not be so relevant because we're wanting to resect that anyway. But I think from my perspective, and this is a purely subjective kind of what do I think is most successful, and I know that other people might have other ideas, but I really am a fan of combining different biomarkers. So generating a detector who can tell you about epileptic spikes, sleep spindles, sleep stages, and HFOs at the same time, and then using a cluster algorithm to say this area is most likely abnormal because you have all your HFOs linked to spikes, and this area is mostly physiological because you have 80% of your HFOs linked to sleep spindles or slow waves, I think could be a good way to do this. And then, of course, the best way would be if you can, for a clinician, if he doesn't have to puzzle that out himself, but you would have like the traffic light that I showed you, red, yellow, and green areas, to say, okay, this is clearly abnormal, this is maybe a normal, and this is clearly healthy, right? That would be my vision for the future. Is that going to work soon? It's complicated because what we see when we verify detectors is that they are often working really well on one data set and then not working so well on another. So I think there's a long way to go still.
Great. All right, I think the last question that I think I would like to ask is, because this is a translational webinar, I'm going to invert the question and ask Julia, what do you think the basic science, what is a question that you would ask a basic scientist to do? What is something that you think it's important to address? And vice versa, Laurent, what do you think it's a clinical question that is unanswered and you want an answer from a clinician, let's say?
Well, I'm going to go ahead because I was so fascinated by the data that you showed at the American Epilepsy Society, how to modulate HFO activity, for example, with modulating or stimulating certain areas of the brain. And I think that is something we will never be able to do in the humans, to understand how HFOs contribute to disease mechanisms. And maybe one of the disease mechanisms I'm really interested in as a pediatrician is epileptogenesis. Like, why do 30% of our kids with a neonatal stroke or hypoxic ischemic encephalopathy have epilepsy 5 years later and the others don't, right? So these are things that you can hardly study in the humans, but you might understand from your models. That's my answer.
Great.
My question to the clinician would be about, when we discuss HFOs in people with epilepsy, we differentiate the seizure onset zone and presumably healthy brain region, and we say HFOs outside of the seizure onset zone are likely to be physiological, but they still occur in a brain of a patient with epilepsy, right? So they could be part of the epileptic network. So my question would be, would it be possible to do a study where you record healthy subject with scalp EEG, obviously, and you compare that with people with epilepsy, or you record people after traumatic brain injury, during a period where you think there might be epileptogenesis before the occurrence of seizure, and you see whether there are changes in the occurrence of HFO. So yeah, the overall question is, can we record HFOs in people that are not epileptic, and compare that with people with epilepsy? I think there
Well,
are a
I'm
few
excited now because we are working on that data. So we are going to answer your question.
Thank you. Keep me posted. There is a study by, I think it's the group of Gregory Worrell, where he used intracranial recording for facial pain. I think, this study where they recorded micro seizures. I would be very interested to see whether he also recorded HFOs and how different it was from people with epilepsy, because this patient did not have epilepsy, obviously.
Great. I think these are great insights. I think we may need another webinar to discuss our newest findings, or another workshop that is coming up. Would like to thank you both for your time and your exciting insights, and everyone that attended the webinar. It's a great pleasure to see you and hear your updates. And thank you so much to everyone.
Thank you so
Thank you,
much.
Christos. Thank you.
Thank you.